Hey everybody, we're back again for another AP video. Now that we've had a little bit more of a foundation in chemistry, I hope to not spend quite as long in this video as we do have a lot of ground to cover. But in this video, we are going to look at the molecular diversity of life. We're going to look at all of the different biological molecules that make up living things and that living things need to survive. And this will cover the Biology in Focus textbook, chapter 3. So, without further ado, let's get started. If you can recall from the previous video, it is the electron configuration that determines the kinds of bonds and the number of bonds that an atom will form. So, different groups on the periodic table will form different numbers of bonds, and then depending on the electronegativity of your atom, you can form different types of compounds. Now, what we are really focused in on here in biology is carbon. Carbon is the basic atom used for all of our biological molecule structures. And this atom is important because it can form 4 bonds because it has 4 valence electrons. And that allows us to make a lot of unique and complex structures. So what these 4 valence electrons can do, forming 4 covalent bonds, and this allows us to make very large, complex molecules. Here you can see a couple of different geometries that can be created using our carbon atoms as a backbone, and this allows us to create very structurally stable molecules in addition to them being rather large. Now, this is nothing that you will need to memorize. We have no memorization of geometries in this class. However, it will be important to note how many bonds carbon can make as we look at the structure of our biological molecules. An important thing to note as we look at more complex structures are the existence of what we call isomers. So that prefix iso meaning equal and mer meaning part. So these are compounds that have the same number of atoms and they have the same elements, but their structures are different. So they have the same parts but different structures. Think like, I'm going to go back to the Legos here real quick, is that we had a pile of Legos and we can build like 3 different structures using the same number and kinds of Lego pieces. So those would all be isomers. What is important to note is that when we change something's structure, we change its function. And function here, we are talking about properties. So properties of our molecules going forward are going to be dependent on their structure. So even though we may have the same chemical formula, we may have different properties because our 3-dimensional geometry is different. And another important piece just to keep in mind as we move forward with these more complex biological molecules is that we can add in functional groups. Functional groups are specifically chemical groups that can affect the molecular function without being directly involved in chemical reactions. We have all sorts of different functional groups that can be added to our carbon backbone, and each one of these groups has a specific function based on its structure. So let's look at a couple of these. So some of the ones that you will come across, now it's not necessarily important to memorize these functional groups. However, we should be able to recognize the type of properties they have, so whether or not they're likely to be providing charge to a molecule. So here we have a hydroxyl with an oxygen and a hydrogen, a carbonyl, which is a double bonded oxygen to our carbon, a carboxyl, which is a carbon double bonded to an oxygen and also bonded to a hydroxide, and an amino group, which is our nitrogen bonded to two or more, if we're talking about ionization, hydrogen atoms. Now, all of these will come up as we look at the structure of our different biological molecules, and they help to determine some of the more complex geometries that exist within those molecules. A couple more to tag on here. These are our lesser groups. Well, methyl's not. Methyl's used quite a bit. But the sulfhydryl group, we really only see that in some of our amino acids, one amino acid to be particular, and then our phosphate group, which pops up in a couple of important places, like our phospholipids that make up our cell membrane, and in our DNA and RNA, our nucleic acids. And one of these molecules that plays an important role in how organisms function is ATP, cellular energy. ATP stands for adenosine triphosphate. Adenosine is a nucleotide, so it's a component of a nucleic acid, and it has 3 phosphate groups attached to it. I'm going to pause here. Okay. And so what's important about ATP, why we use it for cellular energy so often in living systems, is because there can be a great deal of energy stored within the bond between the second and the third phosphate group. And when we break that bond using water through the process of hydrolysis, we can release a lot of energy that the cell can use. You're going to see ATP pop up quite often in the future. I know we spent a lot of time in the previous video discussing water and its structure and how that structure determines the properties that allow living organisms to live on Earth. But besides water, our, second most important compounds are organic compounds. These are our macromolecules, our biological molecules. So these molecules are critically important to the survival of organisms and make up a rather large portion after water is removed from a living system. And these come into 4 classes. We have our carbohydrates, our sugars, our lipids, which are our fats and oils, our nucleic acids like DNA, RNA, ATP, a whole lot of different ones, and proteins. Now, we should keep in mind that the prefix macro means large. So these are large molecules. These are complex structures made up of many different parts. One of the things that you will be asked to remember as we go forward with AP is that all of our organic molecules contain additional elements aside from carbon, and that helps us determine which type of macromolecule we have. So for instance, carbon is going to have carbon, hydrogen, and oxygen, and this is typically in a 1 to 2 to 1 ratio. So the ratio is controlled in carbohydrates, which differentiates it from lipids, which have the same 3 elements, sometimes phosphate when we talk about our phospholipids, but there's not a precise ratio of our elements in the lipid class. Next, we have proteins. Proteins add in nitrogen with that amine group attached to the central carbon. And these also have a variable group that we call R. R is not an element. R is a stand-in for the different attachments that can be made to this base structure. And sometimes that R group can contain some sulfur. Finally, our most complex of the basic structures would be our nucleic acids. Nucleic acids contain carbon, hydrogen, oxygen, nitrogen in that nitrogenous base, and phosphate in the phosphate group. Memorizing the different types of elements that can be found in your different macromolecules will be important when it comes to understanding their larger structures and differentiating between these molecules. If you can recall from Unit 8, all of the elements that are required to build these macromolecules are cycled through the environment. We looked at the carbon cycle, the water cycle, the nitrogen cycle, the phosphorus cycle. Everything is cycled, which means that we can take it in different forms, we can break it down, and we can use it to build new macromolecules. An important thing to note is that our macromolecules are what we call polymers. Poly meaning many and mer meaning part. So they are large structures that are made of many repeating subunits that we call monomers. Mono meaning one. So you can see here that we can take two monomers, we can combine them together to form a dimer, di meaning two. And three monomers make a trimer, tri meaning three, like a triangle. And then we start losing on those prefixes a little bit. We have an oligomer, which is many different parts that are combined together until we get to our largest structure, the polymer, a large molecule consisting of similar building blocks. When we want to form a polymer, it undergoes a reaction called dehydration synthesis. So let's break that word down because that word tells us exactly what it does. It dehydrates, so it removes water, and it uses the removal of water to synthesize or to make something. So we can take two monomers, remove a water molecule via condensation, and form a covalent bond between those two molecules. This can also be referred to as a condensation reaction, and you may see these terms show up interchangeably when it comes to the AP exam. Similarly, we can break down polymers by doing the reverse of the dehydration reaction. That is, we can undergo a hydrolysis reaction. Hydro referring to water and lysis meaning to break down. So when water is added into this molecule, it breaks the covalent bond between those two monomers, and we get separate molecules once again.
Small molecules, called monomers, are assembled into long chains called polymers by a dehydration reaction. Let's look at this reaction more closely. An enzyme covalently bonds two monomers to each other with the loss of a water molecule. One monomer provides a hydrogen, H, while the other provides a hydroxyl group, OH, and water is formed as a byproduct. This reaction is repeated as monomers are added to the chain one by one, making a polymer. Polymers are disassembled to monomers by hydrolysis. A different enzyme breaks the bond between monomers by adding a water molecule, with a hydrogen from water attaching to one monomer and the hydroxyl group, OH, attaching to the other.
With the remainder of this video, we're going to be focused on the four major classes of biological molecules, and so we're going to spend some time with each of them. What's important to keep in mind for the AP exam, what you're expected to know, are the structures of these molecules. You don't have to memorize them, but you do need to know which elements are contained within each, and the different polymer versions of those, and the functions that these different molecules play. So we will start off with carbohydrates. Carbohydrates include our simple sugars, which we refer to as monosaccharides, and our polymers of those simple sugars, our polysaccharides. Carbohydrates play several functions within the living system. They are a source of stored energy. We will follow carbohydrates later on when we look at our cellular energetics. So when we're creating energy from light and when we are breaking that energy down into more usable forms via cellular respiration. They're also used to transport stored energy within complex organisms, that being the role of glucose and other simple sugars, which can store the energy and can be moved around within our living system. They are also a structural component. The cell walls of both bacteria, fungi, and plants are all made of different types of carbohydrates, which allow organisms to have shape. And they are also used for recognition or signaling molecules that can trigger specific responses within the system. So we will find them often kind of as little flags outside of our cell membrane. They are attached often to proteins to allow different molecules to identify different locations or where they're supposed to go. So let's take a look at the structure of our monomer for our carbohydrates, those being monosaccharides. You may recall from previously in the video, we noted that carbohydrates are composed of carbon, hydrogen, and oxygen in a 1:2:1 ratio. And we classify our monosaccharides based on the number of carbon that they have in their skeletal formula, as well as the location of their carbonyl group. Now, here is a cyclic version of glucose. So we don't have a double bond to that oxygen molecule. However, if we were to look at the straight chain, we were to break apart this molecule into a linear structure, it would gain that double bond with the carbon and oxygen. Now, it's important to identify the location of our carbonyl group because that allows us to determine the directionality of our carbon. And this being important to its structure because the directionality of the carbon and which carbons are bonded to what other structures help us determine the structures of our higher classes of carbohydrates. So we always start with the carbon that is attached to, we start with the carbonyl carbon, and then we move clockwise around that molecule. So here we've numbered our 6 carbon molecules, starting with the carbonyl group, and moving clockwise from that group, we have a hexose, a 6 carbon sugar. As noted previously, we often draw our carbohydrates as linear skeletons as well as cyclic because most of the time when carbohydrates are present in aqueous solution or water-based solutions, they will form that ring. Now, monosaccharides serve as our major nutrients for our cells and raw building materials for other larger molecules. So let's say that we wanted to take 2 monosaccharides and we wanted to combine them together. Well, we would be forming a disaccharide, meaning 2 sugar molecules. Now, through the process of dehydration synthesis, which is common to all of our biological molecules, we can remove a water from these 2 monosaccharides. We can form a covalent bond, which here is termed a glycosidic linkage specifically, and we have a disaccharide molecule. Now, disaccharides are pretty common as well. You may be familiar with a few of them. These include things like sucrose, lactose, and maltose, which show up very often as sweeteners. Now let's say that we wanted to take more than just 2 simple sugars. We wanted to combine them to form even larger structures than our disaccharides. Well, then we would be getting into our polysaccharides. Our polysaccharides are our polymers composed of monosaccharides, and all of our really important polysaccharides are made up of glucose, but it's glucose combined in different ways. So here you can see our 3 major classes. I don't have chitin on this diagram. I think I include it in a later slide. But we have cellulose, we have starch, which has 2 different types of chains, those being amylose and amylopectin, and glycogen. So you'll notice that all of the structures of these polysaccharides are quite different, and so you might be able to surmise that they play different roles within living systems. So we'll start first with our storage polysaccharides. Storage polysaccharides are used to store energy. So in plants, their storage polysaccharide is starch. And you find a lot of starch within the roots of different vegetables. Think like potatoes, turnips, carrots, all of these that grow underground. They're part of the root structure of our plant, and they store all of the extra starch as granules. And so these are very energy dense parts of our plants, and can be easily broken down by our herbivores to gain that energy through the process of cellular respiration. Now, animals do not store energy as starch. Instead, we store energy as glycogen. Glycogen is primarily found within the liver and muscle cells because these are locations where you most often need to break down the energy in order to do basic functions. Think like move with your muscle cells. Now, storage polysaccharides differ slightly in structure from our structural polysaccharides. Our structural polysaccharides are used to provide that rigidity to our molecules. So these are also composed of glucose, but glucose in more of a tight weave. So these are largely insoluble, which means that we're not able to break them down. We find an example of a structural polysaccharide as cellulose, a component of the cell walls of plants, and as chitin, which can be found as a component of cell walls in fungi and as an important structural piece in exoskeletons of some invertebrates. The next biological molecule that we're going to take a look at are lipids. Remember that lipids also have a structure that is based on carbon, hydrogen, and oxygen, but we don't have that nice ratio that we see in the carbohydrates. Lipids are also not true polymers because they're not made of monomers. Instead, we classify what we consider lipids by their non-polar hydrocarbon structure, which means that they're hydrophobic. Now, when we look at lipids, we're going to look at fats and oils, phospholipids, and steroids. Each of these different lipids have differences in structure, which help determine their overall function in biological systems. And they'll pop up quite often throughout our study going forward. So we're going to start first with the basic structure of a lipid. When we look at the structure of a lipid, when we look at the structure of a lipid, we have 2 component parts. Now, these are not monomers because they're not repeating subunits. Instead, we have a glycerol base, which a glycerol is a 3-carbon alcohol with a hydroxyl group attached to each of our carbons. And each of those carbons can be attached via dehydration synthesis to a fatty acid. Now, fatty acids can be very long. They can be very short. We can essentially attach carbon after carbon after carbon for as long as we want or as long as we're able. Now, each one of these fatty acids are attached to the glycerol molecule by that dehydration synthesis reaction, and we form covalent bonds in the form of ester bonds. Now, you'll notice here, unlike with our carbohydrates, that every time that we make one of those bonds, and you'll see here we have 3 fatty acid side chains, that we release a molecule of water for each of those molecules. So we can release a lot of water in the formation of a full fat or oil. Now, you may be familiar with fats as they are the primary function for living organisms, being energy storage. So we can store a lot of energy in each of those bonds within the fatty acid, and so they are primarily used to store energy for the long term, which is why you'll often see them in organisms that need to maintain warmer temperatures. They'll have a good layer of fat. When we look at the fatty acid side chains of our lipids, we can differentiate them based on the number of hydrogen bonds that are present by each of the carbons. So we have 2 major classes of fatty acids. We have saturated fatty acids. Saturated means that we have as many hydrogen atoms attached to our carbon as possible. So no double bonds are present in our fatty acid side chain. However, if we remove a hydrogen and we form a double bond between any of those carbons, we become an unsaturated fatty acid. This causes our fatty acid side chain to kink or bend, which causes the molecule overall to be less likely to be in a solid form. Now, if we take a saturated fatty acid and we convert it chemically to a saturated fatty acid, it is a trans fat. So we have chemically changed it from a liquid to a solid, which is what you often find with substances like margarine. We've taken vegetable oil and we have chemically treated those fatty acid side chains to straighten out and become a solid at room temperature. Now, fatty acids, when we discuss them in AP biology, are going to be most often brought up in reference to our phospholipids. Phospholipids being the structural component of our cell membranes. And here you can see that we have the phospholipid structure, which is a glycerol base with 2 fatty acid side chains and a phosphate group. The phosphate group is charged, so this creates what we call an amphipathic molecule, meaning that different regions of the same molecule have different charges. That negative charge in the phosphate group is going to cause it to be hydrophilic, so it likes water. And the non-charged hydrophobic nature of the fatty acid side chain means that it doesn't like water. This is very important when it comes to the building blocks of our cell membranes, as we can create these regions of different water affinity. Another lipid that we'll see come up again when we talk about the cell membrane are our steroids. Steroids are important components of animal cell membranes, and they help to provide stability to different structures of that cell membrane. Here you can see that steroids have a slightly different structure when it comes to our lipids overall. We have a fatty acid side chain, and that is attached to some fused rings. Now, cholesterol is an important steroid to note. That is the one that appears in our cell membranes. But steroids are also very commonly used throughout the body. Not that the other biological molecules aren't important, but by far the most important biological molecule in living systems are our proteins. Proteins are important because they play so many roles. They are literally the workhorses of our bodies. Now, a protein is composed of one or more polypeptides, peptides being the polymers that are built up of our monosaccharides, in this case, amino acids. So you can see here on this slide all of the different biological functions that our proteins can have. They act as enzymes, hormones, receptors, defensive mechanisms, storage, movement from our contractile and our motor proteins, provide structures, and allow the transport of molecules through our cell membrane. Now, all of these different functions are going to come up throughout the rest of the year. So we're not going to spend all too much time getting into the nitty-gritty of each at the moment, but just keep in mind that proteins will come up quite often. But let's take a look at the basic structure of our monomers for our proteins. These are amino acids. Amino acids have a base structure with a central or alpha carbon that is attached to an amine group, a carboxyl group, and a side chain. Now, this side chain allows us to have 20 different types of amino acids, and these side chains each have different properties. When we look at an amino acid, it's important to note that the directionality of our amino acid comes from its amino and its carboxyl group. So we read in amino to carboxyl direction. I find it is important to note that you can read this as we're going from A to C. So amino group with our nitrogen and our hydrogen, and carboxyl group with our carbon and our oxygen atoms. Now, just to introduce you guys to the different types of side chains that we have present in our amino acids, you can see here that we have a wide variety of structures. Now, all of these different structures provide our amino acids with different functions, different properties. And these are going to be important when we get into the higher levels of protein structure, because these groups all play differently with each other and with other molecules outside of the protein. So they allow us to determine which parts of our proteins are hydrophilic, which parts are hydrophobic, which parts like to group together, and which parts may be used for structural stability. Now, amino acids can be linked together through dehydration reactions to form our peptides, polypeptides being many amino acids in a row, and the removal of water in this case forms a covalent bond known as a peptide bond. Each of our polypeptides has a unique sequence of amino acids, and the sequence of amino acids determines our overall structure and therefore our function. So when we talk about the structure of a protein, we're dealing with many different levels of that structure, and it's very important that our proteins retain this structure if we want them to function as intended. Now, proteins, being three-dimensional structures, get their function based on the different levels of folding that occurs in their formation. So here you can see that we have four different levels of structure: primary, secondary, tertiary, and quaternary. There are some proteins that only have tertiary levels of folding, but each one of these levels is determined by different interactions of different parts within our amino acid sequence and overall polypeptide structure. And it is very important to note that the structure helps us determine the function of the protein. We have so many different interactions that a breakdown of any of these can cause our protein to no longer function as intended. Because proteins play so many different roles within living systems, their structures are relatively diverse, but those structures are determined by a few common levels of structure. So we'll start off with primary structure, which is just the sequence of amino acids, so the order in which our amino acids are combined together by dehydration reactions. Once we get past primary structure, we start to get some of this folding. Proteins can either form into coils, which we call alpha helices, or folds, which are beta sheets, and these are caused by the interaction between non-neighboring amino acids. We get hydrogen bonds that form between the amine group in one amino acid and the carboxyl group of another amino acid that is not directly beside it, so non-neighboring. From our secondary structure, we move into tertiary structure, and this is determined by our side chains. So those variable R groups, because we have all different types of R groups, we can get all sorts of different interactions at this third level of structure. These can include things like ionic bonds, so interactions between different charges on our side chains, hydrophobic interactions, hydrogen bonds, and probably one of the most important are disulfide bridges. So disulfide bridges being combinations of our cysteine groups, which help to provide stability to our overall polypeptide structure. Now, like I previously mentioned, there are quite a few proteins that only go up to tertiary structure, but we do have the potential to combine multiple tertiary structures together to form quaternary structures. So for our larger polypeptides, the quaternary structure is where we get those multiple subunits coming together. And you should probably know by now that since I mention it quite a few times, that structure determines function, and therefore if we change the structure of a molecule, it really does have an impact on how well that molecule is able to function overall. So one of the really good examples of changes to our protein structure having disastrous effects are with our hemoglobin molecules. So hemoglobin, if you have sickle cell anemia, you have a mutation in your primary structure. So we have a substitution, a glutamine for a valine, and this causes our proteins to have different structure overall, which causes our red blood cells to be misshaped, shaped more like crescent moons than the normal circles that we see within our bloodstream, and that causes it to be more likely to aggregate or bunch together. This also decreases the ability for our hemoglobin to carry oxygen from one place to another within our bloodstream. So sickle cell anemia can be devastating for some individuals with that mutation. But aside from just mutations within the structure, there's also an important note that our proteins can be broken down based on the environmental conditions that they exist in. So we have these physical and chemical conditions that can disrupt the secondary and tertiary structures of our proteins, which lead to misshapen proteins that no longer function as intended. This is what we call denaturation. So denaturation can come from changes in pH, the solute concentration of a solution, the temperature, and a whole bunch of other factors. And once we disrupt the structure of our protein, it essentially becomes biologically inactive, so it no longer works as intended. Now we're coming to the end of our discussion on biological molecules, and we've got one more to look at today, that being our nucleic acids. Now proteins, their structure is encoded within the nucleic acids that organisms maintain within their bodies. So we can have nucleic acids that store, transmit, and express our genetic information. And we're going to look at nucleic acids in much greater depth later on in the year when we get to gene expression. But it should be important to note that we have two major classes of our nucleic acids, those being deoxyribonucleic acid, or DNA, which stores all of our genetic information and transmits that genetic information to our cell. And then we have ribonucleic acid, which is RNA, which can be used as an intermediate, so a messenger essentially, to take the information that's stored in our DNA molecule and build our proteins from it. So looking at the individual structures of these two separate classes, they both consist of nucleotide monomers, and the base structure of a nucleotide monomer is the same for both of them. So we have this pentose sugar, a 5-carbon sugar, and that 5-carbon sugar is attached to a nitrogenous base, so a base that contains nitrogen, and a phosphate group. Now, the nitrogenous bases that we see within our DNA and our RNA molecules can come in two different forms based on the number of rings that they have. So we have in our nitrogenous bases two separate classes, our pyrimidines, which are nitrogenous bases with a single ring structure. I tend to remember which of these are pyrimidines because you have the Y present in both thymine and cytosine. And we have purines, which are our nitrogenous bases with a double ring structure. Now you'll notice here that we have 5 different nitrogenous bases, and the nitrogenous bases present in our DNA and our RNA molecules does differ. So in DNA, we have adenine, guanine, thymine, and cytosine, but in RNA, thymine is replaced with uracil, which has a very similar structure aside from one extra carbon on that not pentose sugar, on the nitrogenous group. Now, like all of our other biological molecules thus far, we can combine individual monomers together through that dehydration synthesis reaction. In this case, we are forming a covalent bond called a phosphodiester bond. And hitting back to that directionality of our carbohydrates, it is very important to note that our nucleotides are combined in a 3 to 5 prime direction, so that the 3 prime of our carbon is attached to a 5 prime of our new carbon molecule, so it attaches in that specific direction. This will be important when we get to gene expression later on in Unit 6, and I will be sure to emphasize it again when we get there. I've highlighted a couple of different important distinctions between DNA and RNA, but these are things that you will want to remember. It will come up on the exam. So DNA is a double-stranded made up of a deoxyribose sugar attached to a nitrogenous base and a phosphate group, whereas RNA is single-stranded, allowing it to create many different types of structures aside from just the helix. And it contains a ribose sugar, so we have an extra oxygen there on our carbon ring, and that is attached to our nitrogenous bases and our phosphate groups. Looking a little bit more closely at our DNA molecules, recall from the previous slide that DNA is double-stranded and it forms a double helix. Essentially, we spiral around an imaginary axis. And we have two major parts of this helix. We have the backbone, which is composed of our sugar and our phosphate group. And then our nitrogenous bases are held together kind of like rungs on a ladder by hydrogen bonds. And the number of hydrogen bonds that are present between each of these nitrogenous bases is determined by which combination we're doing. So each one of these strands is what we call anti-parallel. So they run in opposite directions. This allows us to keep our molecule even on both sides. And it allows us to use what we term complementary base pairing. So each one of our strands of DNA can be used to create another strand of DNA. And one of the more important pieces to remember, this will come up multiple times, are which nitrogenous bases pair with which other nitrogenous bases. So adenine in DNA pairs with thymine, apples in a tree, and cytosine pairs with guanine, so cars in a garage. Now, each one of these complementary base pairs will allow us to regulate and replicate our DNA in a fairly orderly fashion, and like I mentioned previously, we will cover that in so much more detail later on, but it is important to note for this first exam the different structural aspects of our DNA molecule. Ooh. Okay, everybody. I know that was a lot. I have included links to individual videos on each of the biological molecules if you feel like you need a little bit more assistance with one or another. And to keep this video from being as long as the last one, I didn't quite make it. We're still about 5 minutes short, but I hope that you guys gained a lot of new information from this, and I will see you in the next one. Bye.